So the ZUMA-12 is a single-arm phase 2 trial where we evaluate Axi-Cel as part of first-line therapy for patients with large B-cell lymphoma. Patients with high-risk large B-cell lymphoma defined as either double hit or triple hit lymphoma, or large B-cell lymphoma patients with high IPI score of 3 to 5 have historically had increased risk of relapse and decreased PFS and OS with standard chemoimmunotherapy. So it is in this context we evaluated the value of Axi-Cel in a study involving 40 patients. To be eligible, these patients had to have high-risk large B-cell lymphoma based on histology with double or triple hit, or large B-cell lymphoma of IPI of 3 to 5, but also, they had to have a positive interim PET scan with a dual score of 5 after 2 cycles of standard chemoimmunotherapy. So once these patients were enrolled and underwent leukapheresis to generate the product, they could receive optional non-chemotherapy bridging. And then once the product has been generated, receive the standard Cy/Flu conditioning followed by a single infusion of Axi-Cel. And the primary endpoint for the study was complete response rate. So of the 40 patients enrolled and treated on this study, we observed an overall response rate of 89% and a complete response rate of 78%. And after a median follow-up of 16 months, 73% of the patients remain in ongoing response. In terms of safety, we observed that this therapy is quite well tolerated in this first-line setting, and no new safety signals have been observed with Axi-Cel compared to prior reports with this agent in third-line setting of large B-cell lymphoma patients. We also compared the product profile in patients with ZUMA-12 in the first-line setting versus the CAR-T product profile treated in the third-line setting with Axi-Cel. And we found a higher frequency of CCR7 positive, CD45RA positive T cells in the ZUMA-12 patient CAR-T products compared to ZUMA-1. And this associated with a higher expansion in vivo post-infusion, and which has previously been correlated with better clinical outcome. So overall, I think this study demonstrated that Axi-Cel may benefit patients with exposed to fewer lines of therapy and is potentially effective for patients with high-risk large B-cell lymphoma in the first-line setting. So at this time, we are in discussions to potentially consider a randomized trial to evaluate Axi-Cel in the first-line setting, especially in patients with high-risk large B-cell lymphoma, to compare it head-to-head with standard chemoimmunotherapy.